Let me introduce Dr. Walston. He is the Raymond and Anna Rubin Professor of Geriatric Medicine, the Deputy Director of the Division of Geriatric Medicine and Gerontology. He received his medical degree from the University of Cincinnati and completed General Internal Medicine Residency and Geriatric Fellowship at the Johns Hopkins University. His research interests have focused on the identification of physiological and molecular underpinning of chronic disease, variety, and late life vulnerability. This research has resulted in an internationally recognized portfolio of variety research that has been instrumental in helping uncover important physiology and molecular changes that contribute to the late life vulnerability. Dr. Walston leads the NIA-sponsored Johns Hopkins
So our goal in
American Independence Center and is the PI of several other grants that focus on inflammation and on translation uses for molecular discovery. In addition, he is a co-founder and the co-director of the Biology of Healthy Aging program, where he provides aging research and career mentorship for junior faculty, fellowship, and graduate students. He has over 200 publications in medical literature and served as the American editor of the Oxford Textbook of Geriatric Medicine. He has won numerous prestigious awards for his research, including the Bouquet National Leadership Fellowship and the Grand Award for Biological Aging Research. Today, the video will give us a talk titled as the New Studies of Biomarkers of Resiliency in Older Adults. Let's welcome Professor Walston and watch his video.
Good morning to my friends in Taipei. I'm so sorry I couldn't make it this year to the meeting, but hopefully this will give you some great insights into the biology of resiliency that we've been working on and lead to future collaborative efforts and working together. As I mentioned, Taipei is one of my favorite cities on the planet. Last year's meeting was absolutely amazing. I hiked up to the top of Elephant Mountain and still have these amazing pictures of that. So in any case, thank you for the invitation, and I will get on with our presentation. So our goal in geriatrics is to help older adults thrive and maintain resilience, high function and purpose. And I put these couple of pictures in here. Queen Elizabeth, shortly before her death, 3 days before her death, met the new prime minister. And the fact that she was nearly 100 years old and able to do that is really something that we want to do. She was, responded well to all sorts of treatments over the years and was quite resilient and cognitively intact. And then you all in Taipei certainly know Nancy Pelosi. She was an 82-year, she's still in her 80s, but she was very vigorous in getting across the planet, landing in Taipei, and just her activities in politics, whether or not you agree with them, she just has been incredibly resilient, and her ability to cognitively fight and physically, functionally do very well is quite amazing for somebody who's now in her mid-80s. So what older adults want is to prevent frailty and preserve resiliency. This is some of my relatives that lived in Arizona until they were about 100. They always told me they wanted to keep high energy levels, have good health. They wanted to have an ability to bounce back when they got sick. They wanted to live by themselves, not with their daughters. They wanted clear thinking with no dementia. And they wanted to have enjoyable social contacts and meaningful activities. So when we think about older people, there is this huge kind of difference. Most kids are kind of the same, but even by teens and 20s, you start seeing a difference in really how people are functioning and functional capacity. And a subset of older people do quite well, often sort of this resilient subset, and other people become disabled a lot earlier. and become frail and lose their independence. So we've always been interested in trying to find the biological differences between these groups because we'd like to be able to push this orange line up further. So the talk today will be built on biological frailty research because I think that was our baseline for beginning to study resiliency a few years back, and the biologic conceptualization of physical resilience and what we're working on right now. The spring study and the biologic characteristics of resiliency are what I'll report on. We're just finishing this study up. We haven't quite done all of the follow-up, so we don't have a huge amount of biologic data, but I will tell you about what we were measuring. And once we have the phenotypes of resilience in these three clinical studies, we'll be able to report those to you. So I just wanted to go back quickly to this conceptualization of frailty. This is a paper we published several years ago, and there are two major concepts of frailty. One is physical frailty, the one that we developed at Hopkins many years ago, and it has these characteristics that we see in older community-dwelling older adults. It's not easy to measure in somebody who's in a nursing home or whatever, but these are people who are out still living in the community, and if they have if they meet three of these criteria, 3 to 5, then they're in the frail category. 1 or 2, they're in the pre-frail, and if they have 0, they're robust. This is one from Ken Rockwood and the group in Canada, deficit accumulation frailty or frailty index people use. And usually it's just the summary of all sorts of things that are wrong with people, right? So if they're disabled, they are incontinent, they have cognitive decline, it just adds up and adds up. And the more points they have, the more frail they are. So conceptually, it's sort of apples and oranges. We found it difficult to measure biologic change in these groups because there's so many different ways to get to a high number of frailty points. Whereas this one is a little more consistent. If people are meeting some of these criteria, they're still ambulatory, community-dwelling older adults. We can find differences, and the same I think is true for resiliency measure. So you've seen this before. This is our slide of physical frailty and the complex pathway that gets there. And in this slide, I called it physical frailty, vulnerability, and lack of resiliency. So out here, we know that people who are frail go on to have these poor outcomes. But over the years, we've looked at these biologic changes, and you see physiology here with chronic inflammation, increased HPA axis activity, increased sympathetic nervous system activity, increased angiotensin system action, low abnormal energy production, and altered anabolic hormones. These are kind of the physiologic changes that we think are the major drivers of some of these muscle changes, nervous system changes that lead people to be frail. We think they are also critically important in resiliency, and I'll talk about that in a little bit. Importantly, there are many triggers that drive this. First, molecular aging. Increasingly the sort of hallmarks of aging. We've been thinking about this for 25 years, but luckily the field has really taken off, and hopefully we can apply some of the more biologic findings to these more clinical translational studies. But you see here like mitochondrial decline, DNA methylation, necroptosis, senescent cells, autophagy, etc. are probably driving some of these things. Not all of them, but some. Certainly the energy level of mitochondria. We know that there are gene variations, environmental changes that activate these things, terrible stress, injuries, things like that can be bad. And then finally, the disease states we're on there. Sorry, I advanced a little early. But this slide shows what we call clinically apparent versus invisible measures of frailty. And if you see a frail person in clinic, we know sort of their physical presence as the exam that I mentioned. But we also often see low albumin and cholesterol, some mild anemia, increased clotting factors. Those will show up in labs sometimes and are not quite a hallmark of frailty, but not uncommon in frail older adults. So you see that in clinic. But under this line, there are all sorts of biologic changes that are happening. And we've been trying to measure them and have had some success, especially in big epidemiologic studies over the years. and looking at measures of inflammation. We just published a paper in Geroscience about TNF-alpha receptor 1 being a really good marker of chronic inflammation, really stable one. Cortisol and HPA axis stuff tends to be up. Activated autonomic nervous system, again, these stress response systems. Hormones, glucose intolerance, these are kind of physiologic things that can be measured in people in labs, but often aren't, just because we don't exactly know what to do with them. But they can be hallmarks of frailty and probably of the lack of resiliency. And then down here, that same sort of aging-related change that can be measured, but ultimately at this point, they're not really very translatable up into patient care. So conceptualization of frailty and resiliency, we know there's some physiologic overlap. This is, in resiliency, we often look at how people are stressed and how they recover. So, if people get a big cut or a fall and are kind of briefly injured, oftentimes they'll pop right back, especially young people. They just really quickly come back to normal. And the same, another stressful event, people will kind of roll downhill and get up. But as people age, they tend to be less responsive to recovery, right? Less responsive to these stresses, and they don't recover as quickly. So this is just a line that shows these things kind of falling. And when people get to be frail, often they are super vulnerable. They have a stressful event, and they just don't really recover. They never get kind of back above, and they could actually die from their consequences. So the theory of resiliency is such that we know that there's a maintenance of complexity, homeostatic regulation. These are kind of big biologic themes with lots of sub-pathways kind of embedded within them. And this leads to an overall systemic integrity and ultimately supports resiliency or ability to respond to stressors. So, it's different than many chronic diseases. You can't quite measure 2 or 3 lab things and say, oh yeah, yay, we know what's going to have who's going to be resilient or not. I think many of these are going to be dependent, the measures that we're looking at, on the systemic integrity, right? So how these systems respond in the whole body is going to be super important. So the theory of resiliency goes back quite a ways. The same people who helped us think through frailty back in the 90s, Buchner and Wagner, it really stated this: resiliency is the ability to rebound from stressors and a capacity to withstand stress. So that's how we define it in older adults. It is a maintenance of complexity. Lou Lipsitz and others have written about that. Reactive tuning, and the more loss of complexity in our ability to respond to stressors, the more vulnerable people are. And then homeostatic regulation, we talked about that. And again, this has all been informed a lot by our scholarship on frailty, because we know that frail people often don't have these things. They tend to kind of come apart. So that's why I think that it's been very helpful to us to get into this field quickly and are about to make important findings. Our colleagues at Duke have also been very outstanding at thinking this through. And you see here, there's a resilience is this dynamic response, right? And stressors, things like illness, injury, surgery, psychosocial things will cause some kind of response in a system. And if you're resilient, you just kind of bounce back after a while. And if you're not, you'll have different kinds of outcomes potentially. And then there's also this pre-stress reserve, these areas, which are probably important in kind of informing what this resilience looks like. So this is some of the early hypotheses that develop resiliency. And so several years ago, we got a major NIH study of the physical resilience in aging called the SPRING study. And basically what we're doing is characterizing resilience to physical stressors in older adults. It's a dynamical physiologic systems. And you see, we have 3 studies, and I'll talk a little bit more about them. 3 stressful studies to older adults that we measure. Great team, biostatisticians, clinicians, just everybody from immunologists, on down the line. So it's been an orthopedic surgeons and anesthesiologists, oncologists. So they've been fantastic in getting this. So the three stressors that we're looking at is knee replacement surgery, initiation of hemodialysis, and bone marrow transplantation for hematologic malignancy. So each of these are, common procedures that happen in a subset of older adults. They're different kinds of stressors, but we're hopeful that by looking at these three procedures, we can find people who are resilient and not resilient, and then identify the biology that's different between these groups. So this is just a quick slide that shows, like in rebound, bone marrow transplant. We get a referral, they get a baseline visit, look at all their function, lots of different measures, lots of biologic measures that I'll talk about, and then we follow them up at 1 month, 6 months, and 12 months. And this is their big stressor right here in the middle. For dialysis initiation, people who are just on the edge of needing some kind of dialysis come in, get their baseline measurements done, functional, cognitive, lots of different blood tests. And then they're followed for several months, and then get follow-up visits after the dialysis starts. And finally, the RESTORE study, which is a total knee replacement. We get surgical referrals, many baseline visits, many come in before they get their surgery, 6 weeks before. Lots of physiologic and physical tests that we'll talk about. And then we follow them up for about a year. So, we're very close to being done now. We have a couple patients left in this one and a couple patients left in that one. And then we can do much of the biologic measures that I'm going to tell you about now. We have some, but we can't quite say who's resilient or not until these 12-month visits are done. So this is our conceptual model of the biology of resilience. So we call it resilience capacity, right? So there's an ability to respond to these big stressors. And this is kind of what happens, right? So say somebody gets knee replacement surgery, they get anesthesia, they get all sorts of maybe trouble during surgery, and then they get their surgery and see how they respond, right? So in the end, we can, over the weeks and months and years, we can see if they develop any of these kind of phenotypes and their outcomes, right? So the subset will be resilient and do great. Another subset will be kind of neutral, not really do very well. And then another subset will probably do poorly. And so that enables us then to look at the good subset, the resilient subset, and look what their biology is and see how it compares to the other ones. So importantly in this study, we have utilized a number of areas that we think are critical to contributions to resilience. So, for example, these dynamic stimulation tests, this goes back to that systemic override ability to respond to stressors. So before people get these stressors, we're measuring glucose tolerance tests, ACTH stim tests and cortisol for the HPA axis. We take immune system cells and stimulate them with LPS, which gives us an ability to determine the strong or weak response of these cells. And then heart rate variability is more of an autonomic nervous system measure. So before anybody gets any of this, we have a very good sense of what's happening with these systemic responses through these sort of minor dynamic stimulation tests. We also measure a host of aging-related things, senescent cells. We're looking at epigenetics, metabolomics, cell-free DNA, and a bunch of different hormones and blood tests. So cytokines and other things at baseline that are potentially very important in predicting who's going to be resilient and who's not. Many people have thought about this over the years. This is again from Matteo Cesari, and talks about which biomarkers are associated with physical resilience. And you see here, senescence, inflammation, mitochondrial function, skeletal muscle metabolism. We've picked up most of these measures, probably a little bit fewer on the muscle piece. And then you see what's low resistance. So these are kind of hypothetical measures and things that we're actively finalizing in the next couple months. So again, I mentioned these measures. I think they're super important. They're kind of core of this sort of systemic response that we see. So again, we measure these before people get their actual procedures, right? So we see how they're going to respond to sort of minor stressors. And we hypothesize that those who do not respond well are going to be less resilient. And those that do respond well and kind of pop back up to normal are going to be far more resilient in their outcomes after their clinical procedures. We know that aging-related contributors to resilience have capacity in the neuroscience domain. We know immune systems in frailty and all sorts of things. We can tell that all sorts of disease states, these really make a difference, and people who have chronic inflammation and altered T-cell phenotypes often do worse, kind of in recovery periods or all sorts of things. So, these are certainly part of that. We're looking at the physical properties of cells, not just the senescent ability, but also looking at senescent changes, but also how they move. We do have a very good collaborator now who is very expert at watching these cells move and measuring the differences between frail and non-frail, and also he's going to do that then in our resilient versus non-resilient subset. So finally, we're in the final part of doing changes in these other kind of markers of. Let me see if I can get back one. Other markers of resilience. I won't go back into it. I'm running out of time here. So, but I did want to do one characteristic that I think has been a Johns Hopkins thing for many years. GDF15 was actually worked on many years ago by Se-Jin Lee here at Hopkins. You see this publication from 2000. This has become a big favorite in geriatrics recently, and it gets to be induced following injuries and other things. And we think it's going to be a very strong, single molecule predictor of resilience or lack of resilience. And I'll just show you some quick data from a subset study that we've done on frailty. Fangyu Liu, an outstanding fellow, has published one paper on this and is about to publish another. But you see in the frail subset in proteins, this GDF15 is way up at the top in pre-frailty, and also pretty far at the top in frailty. So we think that it's going to end up being quite important. There's other important proteins here too that may end up helping us predict. You see this TNF-alpha receptor again that seems to pop up all the time. So we'll see relatively quickly which of these things are promoting resiliency or backtracking resiliency to stressors. So what does GDF15 do? It sort of circulates centrally and regulates appetite. It regulates macrophage activity with metabolic activation and alterations, and then it sort of regulates this. Again, because we don't have the final resiliency results, we know there's a subset of people who did very well, a subset who did very poorly, but we can't really do the analyses until we're completely finished. So this is just a graph of the sort of measures of this in sort of healthy middle-aged men and women. In our spring participants, you see, it's they're older, and it's kind of pushed out to higher levels out here in the older people, which is not unusual. So if you look at our three studies, you already see some difference in this. First, in rebound, which is the cancer subset, you see that there are a lot of people who are kind of in this normal range, but there are a subset that have high levels here, and we would again hypothesize that these are going to be the non-resilient people, the people that may well die within a month or 6 months, or never really they may be cured of their cancer, but they don't recover functionally or cognitively. In the ready or dialysis subset, you see the levels are much higher, which is probably because it's not getting dialyzed or cleared out of their system. these are before they have their dialysis, so it's kind of floating around. But long term, these folks who are waiting for dialysis are at very high risk of all kinds of things. So this particular molecule may be driving some of that. And then finally, Restore, which is the knee replacement. You see, it's probably not as important, interestingly, right? So you see, like, a lot of these people, they're older, they're in their 70s, but they have, pretty good levels. And I would predict that these are the folks that are just kind of doing quite well. And these few are the ones that didn't do very well after their surgery. But we'll find that out very shortly once we get measures done. So I think I will skip this. We've also done a lot of metabolic measurements, with our we know that energy changes are super important in frailty and drops in energy, glucose intolerance. And so we're beginning to delve into the very deep biologic changes that are happening. And you begin to see in these frail subsets, things like ketoglutarate, malate, fumarate, some things along this TCA cycle are really important, and we're gonna do a lot of work there to determine if there are some sort of roadblocks here that are dropping energy levels or something blocking this, so that people who have these elevations are less likely to be able to recover because of their the changes in how they make energy and use energy to repair their tissues. Finally, aging-related contributors to resilience capacity. We are actively measuring senescent cell markers in the knee tissues with the colleagues at Mayo. So hopefully next year we'll have some really, really important findings there. Cell-free DNA, which activates immune system changes. We're going to look at that. Mitochondrial energy and metabolomic measures. I showed you a little bit of that, but those are really going to be a big piece of what we're doing in these age-specific changes. So finally, what's next in the field? Again, we're going to identify resilient outcomes in knee replacement, new dialysis, and bone marrow transplant. They're very different stressors, and the subjects who come in for these studies are older, but they're also physically probably different in a way. These folks have had more chronic illness. These folks have an acute illness and are generally in pretty good shape if they're going to make it to get bone marrow transplant. And then knee replacement, there's a huge variability in people who are obese, people who have rheumatoid arthritis, all kinds of things that have gotten them to that place. So it's a lot of variability here. And so we're going to look at the relationship between the baseline biologic, clinical, and dynamical systems, as well as psychological variables with these resilient outcomes. So, some people do well, some people do poorly, and we can look at the biologic differences between the two. And we'll be doing that basically in December and January. And hopefully next year I can come to Taipei and present a really beautiful amount of data on this. And so we're also going to be developing models that facilitate better understanding of interactions. So some of these things may pop up in all three of these stressors. And that would then be a way to begin to look at biomarkers in clinical practice that may help predict how people are going to do. So that's a super important kind of geriatrics field builder, because sometimes people get terrible injuries and are going to do very poorly. For example, if somebody is going to get a bone marrow transplant and has some of these markers, they would often die very quickly after the bone marrow transplant. But if you didn't do it, they may live 6 months, a year, not do great, but they have a better health span and lifespan. So those are the kinds of things we hope to do in all kinds of different geriatric measurement areas. And then finally, interventions based on biologic discovery that promote that. So, to fix up some of these systemic changes that we think are going to be terrible. And then finally, develop new care plans for individuals who might have had resilience or lower likelihood of good outcomes after some kind of stressful event. So again, identifying differences between older adults and how some might do great and some might do poorly, and we really have to not just throw everybody into the same bucket. I think that we can make some differences through our clinical translational work and biologic aging. So with that, I will acknowledge our NIA-funded Older Americans Independence Center and a huge grant from the NIA to get us to where we are. I think we will do very well down the road with this, and I'm excited about presenting it to you hopefully next year. And again, thanks for the great invitation. I greatly appreciate it, and hope to see you soon. Sorry I couldn't come.
Okay, it's quite inspiring, right? So if you have any question, please send your question by email to him. Okay. Don't ask we too. Okay.